right, last section. Sorry, this lecture is so long today. Just one point of clarification. I think when I was talking about synapsis and chiasma, so here synapsis, I just want to clarify that the synapsis is the process by which your homologous chromosomes pair up as tetrads, right? Chiasma is the actual location or site of attachment for crossing over. Okay, so just to be clear on that. Okay, so we were ending in metaphase one. So metaphase one, again, we have our tetrads, or our homologous chromosomes are paired up in this metaphase one, which is different from mitosis. And so here, basically, you have your tetrads that are lining up on the equator or the metaphase plate. So then in anaphase one, this is where we actually separate our tetrads. So basically, our homologous chromosomes here are separated from one another. So you can see that our sister chromatids, as this says, remained attached to each other. So the sister chromatids for now, the identical copies of one chromosome, are still attached, but we actually separated our homologous chromosomes, okay? So your key here is that in anaphase one of meiosis one, your homologous pairs are separating here. So then telophase and cytokinesis, right? Similarly, you're going to have basically the opposite of prophase, where your nuclear envelopes temporarily reform, right? And then you're also going to, depending on the type of cell, and animal cells will have the cleavage furrow that occurs. And just remember that telophase and cytokinesis typically occur simultaneously. So just FYI. One other major thing here is to note is that at this point, before the second round of division, we are at haploid cells. So these two daughter cells that we've produced after one round of division are now haploid. And the reason for that is because we remember that in the last step, we actually separated our homologous chromosomes from one another, right? So now we have one set on this side and one set on this side. So we actually have just created haploid cells where we started with diploid cells. Now you might get a little confused because you're looking at these like, well, they're still duplicated or attached. And you're right, your sister chromatids are still attached to one another here. But remember, these are identical copies of the same chromosome. So we identify haploid or diploid by the number of chromosomes that you have or the sets of chromosomes that you have, right? So now, since we've separated out our homologous chromosomes from one another, now you have n equals 3 here, right? And n equals 3 here, okay? Whereas here, let me go back to your original. Well, let's just look at this one. In this original cell, your n would equal 3, but 2n or diploid would equal 6, right? So here we had 6 chromosomes, or basically 3 sets, 3 pairs. Now we've ended up with one set of 3 chromosomes in each of our resulting daughter cells. So at this point, we're already at the haploid phase, but we do still need to separate out our sister chromatids from one another. So we need to do one more round of division. So here's just a brief recap, right? So again, in meiosis one, major things that occur here. In prophase one, you have the tetrad formation between two homologous chromosomes, so homologous pairs pair up, and crossing over occurs. So that exchange of genetic information that's going to increase diversity. Okay, so that's really important. The other really important thing that happens here is that in meiosis one, homologous chromosomes separate, and we do end up with 2 daughter haploid cells at this point. So meiosis 2 is super similar to mitosis. Now this should be familiar to you. So here, basically, again, prophase 2. And just to make a quick note, there is sometimes a brief interphase between meiosis 1 and meiosis 2, but regardless of whether an interphase actually occurs, no DNA replication happens. Okay, so between meiosis 1 and meiosis 2, we are not copying any more DNA. All right, so we're at our haploid cells. Now we just need to separate out our sister chromatids, and then we're all set. So no DNA copying occurs. We move into the second round of division. So here in prophase 2, again, same thing. So think preparation, our nuclear envelopes break down, spindle fibers form, centrioles are migrating to the poles. The chromosomes, if they did temporarily decondense, they recondense into chromosomes, right? So they show up. And then you move into metaphase 2, right, where they line up along the plate. Anaphase 2, where your sister chromatids are separating at this point. And then finally, telophase and cytokinesis, we're going to end up with 4 daughter cells total, or 2 daughter cells resulting from each of these daughter cells from the first round. Okay, so again, meiosis 2 follows meiosis 1 without any chromosomal duplication, as I said. So no DNA is copied here. So again, prophase 2, you have your chromatin condensing into chromosomes, nuclear envelope breaks up again, right? So this you're familiar with at this point. Metaphase 2, your duplicated chromosomes, right, are going to line up at the siliquator. And again, your spindle fibers are attaching to those kinetochores in the centromere region, just like we learned in mitosis. Anaphase 2, this is when sister chromatids are pulled apart. So in anaphase 2, it's the sister chromatids or the twins that are being pulled apart from one another, right? So here you can see they've been pulled apart. So again, this is just like in mitosis. Major difference though, you can see here that remember that your sister chromatids now have genetic diversity because of that crossing over that happened in prophase 1, right? And then telophase 2, basically, the opposite of prophase, your nuclear envelopes are going to reform, your chromosomes are going to decondense and go back to chromatin. Again, we will end up with 4 daughter cells at the end of telophase 2 and cytokinesis. Okay, so cytoplasm will divide and we'll end up with 4 daughter cells, which are haploid. So basically 4 daughter cells that are haploid and have 1 set of chromosomes each. So in humans, that would be n equals 23. In this picture, n would equal 3, right? So in this diagram, we have 1, 2, 3. In humans, n would equal 23. So this is just a comparison chart of mitosis versus meiosis. Again, I did provide for you this PowerPoint, so you can just print this out if you would like. But this is important for you to be able to compare mitosis versus meiosis. So understanding the goals and the differences between these two processes are essential, right? So remember that mitosis, the whole goal here with mitosis is to produce identical copies because you need to be able to grow, repair, replace, right? And then organisms that asexually reproduce would use this as their method of producing clones of themselves, all right? Meiosis, on the other hand, this is the production of egg and sperm cells. So we are going to half the amount of genetic information that we start with to produce eggs and sperm because, again, those will combine to create a new human. We want genetic variation. We want diversity. So we want crossing over to occur and gene shuffling to occur. And here is just a diagram, and your book has, let me check maybe the same one. I think it does have the same one. So this is a really good diagram to compare and contrast what's happening in mitosis versus meiosis. So again, in both, you start with a diploid cell, and it says here, this is before chromosomal duplication, right? So you have your N is 2 here, 2N would equal 4 for your diploid, right? Then you have DNA duplication here, and then you can see some of the major differences. So again, in meiosis, we have that tetrad formation and crossing over that occurs, right? Metaphase 1 looks very different in these two because since we have the tetrad form, we need to pull apart our homologous chromosomes to create haploid cells here, and then we undergo another round of division for meiosis 2. You'll also notice that, again, if you compare this parent cell to your mitotic daughter cells, these are identical. So mitosis, you're producing identical copies. In meiosis, look at this parent cell compared to your daughter cells. You can see that you have half the amount of genetic info in each of these haploid cells that you did in the parent cell, right? So we've halved the amount of genetic info. These are haploid, whereas the parent cell was diploid. So again, meiosis leads to genetic variation, and the way that works, right, is of course we have, well, a few things. So genetic variation due to the shuffling that occurs from crossing over. Also, when chromosomes line up during metaphase 1, they line up independently of one another, meaning they're not going to necessarily get into exact order. So this process occurs over and over again to produce all your egg cells or all your sperm cells. And every time that occurs, your chromosomes might line up in different locations. And so then when they get pulled apart, they might end up in different areas. Okay, so they don't line up, in the same order every time. And then random fertilization is another way that we get genetic variation. This is just the idea that you're not controlling which egg and which sperm, unless you're using in vitro fertilization, is going to actually conduct the fertilization, right? So you have basically different combinations. I think I read a stat where males produce, I want to say maybe trillions of sperm, and each one of those sperm is genetically unique from one another, right? So there is no way to reproduce you, right? The only way that you have someone who has the same genetic information as you is if you're an identical twin, that actually identical twins come from the same zygote or the same egg and sperm that were fertilized, right? So independent orientation or independent assortment. So again, this is just that idea that chromosomes line up independent of one another on the metaphase plate. So again, if you think about the possibilities, so this would say 8 million potential chromosome combinations, depending on how they line up, and thus how they're split apart. So again, here's just a picture here for you, just showing that chromosomes, when they line up, right? Possibility A, you might have these chromosomes on one side, these on the other, which would mean these two would be pulled together on this side, these two would be pulled on this side, or they could line up like this, right? And it end up that these two go over here and these two go over here. So you have a bunch of different combinations. Again, that's exponential depending on how many chromosomes you have. So with us, we have 23 pairs. We're going to have obviously more potential for different combinations. So here's just going through what that would look like, right? So you can see here again, depending on how the chromosomes line up at the metaphase plate before they're split apart, and anaphase will affect the combinations that occur in your resulting daughter cells, which would be either sperm or eggs down here. So homologous chromosomes may have different versions of a gene at the same locus. So we talked about this a little bit, but again, that's the idea that if you were to find the gene for coat color, for example, if we're looking at this little rat here, right? Basically, you have the coat color gene and the same locus, the same location on homologous chromosomes, but on one parent chromosome, you might have a gene for black or for brown, and the other one you have for black, right? So again, that could just depend. So again, this is looking at brown versus black, white versus pink. And here's just looking at how you can have these different combinations work out, right? Because the other thing too is that genes are inherited, separately from one another as well. And crossing over can increase the amount of shuffling that you have. Okay, so that is what I have for you. So one other thing that I want to go over, I'm just looking for the picture here. So I'll just use this picture to talk about. But one other thing to note is that when we talk about meiosis in sperm production or spermatocytes in males versus oocytes in females, one thing I want you to remember is that technically 4 daughter cells are produced, whether or not we're talking about meiosis in males versus females. But in males, all 4 gametes, all 4 sex cells are viable sperm, meaning they're functioning sperm. Whereas let's say this is female, if we're talking about a female, only one of the 4 oocytes is an actual egg. So only one of the 4 daughter cells that are produced is a functioning egg. And a lot of that has to do with the idea that the egg is actually the host, right? That gets fertilized, but with the sperm, it has all the organelles and houses all the cytoplasm and all this other stuff. So during meiosis 1 and meiosis 2, basically all the energy kind of goes into one egg, right? The other 3 daughter cells that are produced in females are what we call polar bodies, and they're non-functioning, they're not actual functioning eggs. So if you think about the fact that males produce a lot more sperm than females do eggs, this is why.